Welcome to another episode of the MyData Zurich webinars, and today with a very specific topic about healthcare through better scientific data. Before I hand over to our guest speakers, a few introductory words about this webinar. So this webinar is co-hosted with DIDAS, which is the Organization for Self-Sovereign Identity. I'll make these slides very quick because you will get the presentation afterwards. You can read that at your own time. Nevertheless, since we are MyData, I would like to point out that personal health data is everywhere. And today the focus will obviously be on healthcare data. This is a pretty old picture from MyData, which shows a little bit how the data developed. In the early days, it was very API-centric, so that was the data Stone Age. Then came the data Middle Age, when the data was mostly on centralized servers. And where we want to go with MyData is actually to privacy by design. And that's also the objective of DIDAS and also of this webinar. I want to point out a special construct of MyData. These are the MyData operators, which is basically the fiduciary of the healthcare data, because the systems are very often pretty technical and pretty complicated. So somebody has to do that. And this role is the MyData operator. You find more details in the respective documentation. A few words about the MyData Hub Zurich. We were founded in 2019. We have 3 of us. So I'm basically doing the more organizational stuff. And we have André Gollier and Jonas Berci, who is also on the board of our MyData Zurich Hub. We are present on LinkedIn, so you can follow us on LinkedIn. And we also have a YouTube channel, which you can see here. So all these recordings are available on this YouTube channel with a few playlists with different topics. You can also become a member, so mydata.org/join. So if you want to support the activities of MyData, then you're more than welcome to join the organization. It's a pretty minimalistic fee. And there is also a YouTube channel for DIDAS, which I mentioned before. And there's a playlist health where you can find another series of recordings and long and short versions. Then one heads up what we are working on right now. It's from a DIDAS perspective. It's a communication activity. And we have a few key messages which we want to convey, and we are currently coordinating with other associations, so to make sure we have the same key messages. And then we are planning to reach out to the respective stakeholders, because some of these topics are not yet really fully identified and fully actioned. Follow DIDAS to find more information about this one. With that, we are already on today's agenda. So we are a little bit late. We'll start with Marco Egbring. I'll say a few words about him with precision medicine and drug interactions. Then a short teaser from Ernst Hafen from the Swiss health data space. Then a short wrap up, and then going into discussion. Officially, the end is after 1 hour sharp. I very quickly introduced our guest speakers, so thank you very much to both of you. I keep it short, otherwise the list would be very long with both of the speakers. Marco is the founder and CEO of Epha Health, and he has a very large background in cogenetics. Exactly. So in simpler words, it's drug interactions and interactions with genomics. And he also clinical pharmacologist and data scientist as a role as a CMO. And Ernst Hafen was the former president of ETH and currently the president of the Swiss Health Data Space Association. The floor is yours, Marco. Thank you for taking the time, and you can take over from here.
Thanks for the intro, Peter. And let's start right into it. Pharmacogenetic, and it means basically the influence of genetics on 2 things. It's very important to distinguish these 2. One is pharmacokinetics. This is the influence of enzymes and transporters on drug exposure. It's a little bit formal, but what it means is if you take a drug, it's usually 100% exposure. And then in the stomach, it's soluted in water, and then it goes to the gut lumen where the drug usually is absorbed. And already in the gut, you have sometimes enzymes and transporters which metabolize the drug. Before it actually reaches the liver for the first time, sometimes you have a really reduced, what the expert says, it's bioavailability. So only a fraction of the drug reaches the liver, and then in the liver, you have even more transporters. So you have transporters sometimes taking up the drug into the liver, and then you have a lot of metabolism in the liver, and then you have from the liver sometimes transporter eliminating the drug to the bile or to the systemic blood flow. So we have a lot of transporters and enzymes going on. And depending on other drugs, because sometimes other drugs change or inhibit enzymes. For example, the classical example is grapefruit. Grapefruit inhibits an enzyme in the intestine, and this changes then the exposure. And this can be dramatic. For example, if you have a drug which has, like, before it reaches the systemic circulation, only 15% is left, because most of it is already metabolized. If you inhibit some of the enzymes or the transporters, it's possible that 50% reaches the systemic blood flow, which means it's kind of 3 times the exposure that you would expect in a normal patient, which is quite a difference. It's just like taking 3 tablets. And then often you have this combination where it's not only the pharmacokinetic exposure changing, it's also the pharmacodynamic part. The pharmacodynamic part means, what does the drug do with the body? So what's the effect here? And sometimes you have a genetic variant, which leads to that you have a more susceptible receptor, for example. And then you have a 3 times higher exposure. You have a more sensitive receptor, and this is then the coincidence where something serious can happen to the patient. And this is why adverse drug events are considered to be the 4th to 6th leading cause of death, according to FDA and WHO. So it happens, a lot of adverse drug events, which can be reduced, dramatically reduced, if you understand everything from the patient. If you have all the data from the patient, it's already today possible to perfectly customize much more than we can actually do like 10 years ago. And how does this work? So from genes to proteins, how does it work? You have your DNA, so you have your sequence, and this sequence is, this coding sequence leads to a protein. So it's a chain of amino acids chained together. And if you have changes in the sequence of your gene, it's possible that your protein looks different than the average protein, which leads to a different activity, maybe, of your enzyme. Sometimes it doesn't affect the activity, sometimes it does, sometimes it's only affects the, for example, the induction of like the production of the enzyme. So there are different things that can go wrong. And how does this look really? This is from a patient movie we did, where we tried to show how drugs changes like a transporter in the body. This is the yellow molecule is insulin. So you have transporters, like the purple ones are working, and the darker ones are not working. Okay, so this is kind of what happens. Sometimes it's just happening with aging. Sometimes these different transporters happen because you have a variant. And it's very important for us to know what is to expect if you have a different genotype. And to make it more practical, basically the medicine in the last decade relied on text guidelines. So we had societies write up guidelines and perform studies and try to formulate what is the best approach to treat a patient. And for example, here you see a drug, it's called paroxetine. And if this is given together with another drug, tamoxifen, which is actually a very good drug, which you give breast cancer patients after surgery. And the problem you have is if this paroxetine is capable of inhibiting an enzyme called 2D6, then the tamoxifen doesn't get activated. So it's not working in the body because it's not activated by this enzyme. And it's labeled in the Fachinfo that resulting in increased mortality if you combine these two drugs. But same thing can happen, and it's also listed in the Fachinfo, if you have a poor metabolizer status. So the enzyme is not working because your sequence is different, which leads to a broken enzyme because you change the amino acid, so it's not working at all. So it's called a poor metabolizer. Then it's also not working. So the drug again doesn't work, and you have a increased mortality, which is terrible. And this is just like the extreme cases. But what we have seen in doing a lot of research, for example, with Hasana, is it's usually never an obvious case because medication is very safe. It's always a combination of different factors. And in this case here, often it's not just one drug, which is like a very intense inhibitor of an enzyme. Often it's you have an intermediate inhibitor as a drug, and then you also have an intermediate enzyme activity, which together ends up to being a poor metabolizer, but it's always the combination of different factors. So what have we done so far? We did a lot of work in providing additional clinical intelligence to doctors besides only these text guidelines. We worked with colors and search the drug label information. So if you have a patient and somewhere written in the text of the Fachinfo is mentioned, this is a contraindication, it gets highlighted. The problem is the Fachinfo is a interesting document, but it's not the best document that exists to guide you through a complex patient with different treatments. So we had to find a solution to provide these intelligence. And one way to do it is to extract all the data that's available in studies from the literature. For example, you take all the drug-drug interaction studies and all the gene studies, and now it's only we focus on pharmacokinetic. Remember, pharmacokinetic is the part where the body, due to transporters or the enzymes, changes the exposure of the drug. So basically, these studies look like if you take drug A and we add drug B, the exposure of drug A is twofold increased. So it's a lot of work to go through the scientific literature, but thanks to the large language models now available, it's much, much easier. And what you can do is you can extract all the studies and extract all the values from the scientific literature, and you construct what's called an optimization problem. So you have a mechanistic model. It's basically a digital twin of the pharmacokinetic part of a patient. And you ask the model, okay, listen, I have all these studies. So these are the endpoints. Sometimes exposure changes, sometimes it increases, sometimes it decreases. And please figure out for all the drugs and all the genes, how much is the influence in this particular scenario. So figure out, for example, for paroxetine, how much is the inhibition value for 2D6? We assume because it's already written in the Fachinfo, it's pretty bad. But at the same time, figure out how much tamoxifen is influenced in these studies and how much is the influence of the gene in the pathway. And models, it's called hyperparameter tuning, and you try to optimize the error rate. And the really great thing is if you work on it a lot, you really get a model which actually generalizes to all the clinical studies. And if you look here on the left side, what you see is, for example, you have metoprolol. This is a drug also metabolized over 2D6. And you have different studies. These are all actual studies here. And you see if I have metoprolol and the 2D6, the IM is called intermediate metabolizer. So I have the substance and the gene is kind of reduced in the activity. I have a 1.8 increase in exposure. So it's almost like having 2 tablets compared to the normal patient. You see this for different genes, but it also works in the same manner for different drugs. You kind of figure out what is the influence of all these drugs on these enzymes. And of course, you have to consider different transporters and standard pharmacological values like what's the clearance rate, what is the protein binding, what's the volume of distribution, and you have to figure out a lot of things, but then you can make it work for the model to generalize, which is really great. So you kind of are able to predict most of the studies, very, very good, and you get values for certain enzymes. For example, the model figured out amiodarone. It's like a very strong inhibitor of many enzymes. You can figure out, so in the gut, in the liver, it has different inhibition values. And if it generalizes in this way, you actually can predict studies which you haven't performed yet. And we tested it. The model is really good. We had a project with Novartis where we actually predicted drug-drug interaction studies which weren't published yet. And afterwards, they planned to do them, and they kind of adjusted the way they designed the studies because it's possible to predict a lot of standard substrates for your drug-drug interaction. And here it's just a sample of studies we can predict. And the interesting part here is you can treat other drugs just like certain variants. So this is why it's so powerful and it works, because we have so many data. Why do we have so many data? Because the pharmaceutical companies have to do a lot of research, very expensive research, to get the approval for drugs. They have to show drug-drug interaction, drug-gene interaction to get the approval of the authorities. This is why there's so much data available, which isn't considered if you do a prescription on a daily basis, but the information is there. And if you are able to have these generalized models, it's easy to predict even complex patients, what to expect in these patients. And then often you have the possibility to change the drug, because for most drugs, you have generics available, like different substances in the same groups. You actually can greatly improve a lot of treatments. Basically, where it looks, you can see a patient, get a snapshot, get the medication. Often you need more than the medication, you need symptoms, you need efficacy, you need the problem of the patient. And then you can predict certain adverse events, and you can give them a score. And if you can give them a score, it's just like creating a chess computer. If you can give a certain situation a score, it's possible to pre-compute a better treatment. So without knowing any clinical guidelines, the model is capable of finding better alternatives for a treatment, and which of course has to be validated by the doctor. If we look at the rest of the world, it's really out there already, and it's kind of surprised us. We started now to do drug-gene interactions in Switzerland. We're testing patients. We're working with pharmacies. We have a great partner. It's called JAI, which kind of identifies patient in the pharmacy based on prescription data. If this patient is at risk of an adverse event, then talks to the patient, asks, okay, do you have symptoms? And then if the patient has symptoms, which are very likely to be caused by a drug gene or drug gene interaction, we do genetic testing and we're helping the patient. And this really works great. When we visited Abu Dhabi last year, we were kind of surprised to see, for example, in Abu Dhabi, 50% of the patients, more than 50% of the patients are already completely sequenced. So the complete genetic data has been sequenced, and they use this data to improve and personalize the treatment of their patients, which is kind of incredible. So it's a trend, and it's a very good trend because it doesn't add any really extra costs. It's just like you make your decision more efficient and smarter, and by avoiding a lot of unnecessary patient harm, you actually spend less resources. So thank you.
Thank you very much, Marco. Really very interesting.
Question.
Okay, let's go into a few questions. Then I would like to give Ernst a few minutes for his pitch for his data space, and then for the rest of the session, then we can go into questions and discussions.
So yeah, if I can start with the question, of course, Marco, thank you very much. Obviously, I'm fully aligned with you, and the pharmacogenomics is sort of the first direct benefit that you get from having your genome sequenced or analyzed, and so now 23andMe and everyone has gone into sort of ancestry, because that is the sort of the thing that Americans at least are interested in. But you're getting the pharmacogenomics for free, and not even 23andMe is really offering this. And so if I'm thinking now of our participants in the expedition that claim their own data, many of them would probably be ready to do some kind of genome analysis, especially if it can be done in Switzerland. And so my question is, what kind of genome analysis would be necessary? Is there a particular pharmacogenomics panel that you can run a set of SNPs using PCR, or is a low coverage whole genome sequencing the cheapest? What is your current view on this?
Well, it would be best to have sequencing data, because then right?
Yeah.
This is sequencing is gold standard, but it's still quite expensive. I mean, the cost came down more than everything else. Like if you look at Moore's law, the prices dropped, and you can get like a small device from Oxford Nanopore, which you can plug into your laptop. I mean, it's unbelievable what they did with the technology. But our test is basically an array, which is very similar to 23andMe. But we're trying to customize, so we have a cooperation with Illumina, we're trying to customize the variants for pharmacogenetics. Because like every day you get new papers published, there's a new computational biology group computing some new variants. It should be on the array, and sometimes it's not. So we try to modify and tune the custom array, which is around, I think it's 2 million variants.
Yeah.
But this is important to get better results, yes. But it's not required, but it's like standard practice. Really try to help them to make these array technologies better.
And how much would that cost for a person to do now, such a sort of customized array? Is Illumina offering this, or would you, or is there one of the genome centers in Switzerland specializing on that? And
well, you can test. There are a lot of centers available in Switzerland. We have a special cooperation with JAI and University Basel, where we get our results. But it's a common technology, and the prices depend on how many. I mean, you can get the basic chip from 23andMe for, I think the price is $99, right? So if you want to have increased quality of the array, I'm sure it's more expensive, but we're figuring out the price right now. It heavily depends on, because these machines are high throughput machines. And if you have a robot, you have to get like a lot of patients to make this cost efficient. Once it gets rolling and you get like a lot of patients, you will get pretty good prices, but you have to distinguish between what is reimbursed by the health insurance, what they're paying for, and it's actually very old school what they're paying for. They should completely change the prices, they reimburse the lab, but right now they haven't. And the price you will get as a private person. So what the health insurance is paying is probably higher. I mean, it's not a lot. It's not like sequencing. It's still cheap considering the benefit. But I think for a person, we're just figuring out the price right now, so I don't want to say a number, but I think it will be affordable to a lot of people. And if it's too much above 23andMe, which is 100, it's hardly to justify, right? And you get like 4,000 almost sequencing, so it will be in the range there.
Okay, so you know, as you will see, our salutonauts, and I already count Pete also as a salutonaut, quite adventurous, and so we can request them to do a 23andMe, get the raw data, and then you could provide the service to visualize my. If I send you my 23andMe data, then you can. So as part of the expedition, that would be a new service that we can offer in collaboration with you.
Yes, and I would use my partners actually, because it's quite a network, and they work with the pharmacies. And something also important, my service is not really valuable for patients without any medication. They expect like, help me to stay healthy. Okay, you have to do sports to stay healthy, but once you take medications and you don't tolerate the medication, then I can be of help. And if I can be of help, then it's reimbursed by the health insurance if it's actually there's a high probability that the drug and the gene is your problem is causing the symptoms. So you don't have to pay anything.
Yeah, because you know, I mean, in an actual case, my wife had some heart problems, and she had to see a cardiologist, and she got a stent, and then of course she got Plavix, and no one knew whether it was. So I had to go into the SNPs of her 23andMe data, and they said, okay, yeah, she responds to Plavix, and these kind of things is good to know before you have all kinds of medications.
Absolutely. Right? I have no idea why in Switzerland it's not happening before you start the treatment. No idea, but it should. Yeah. And it's really helping, because a lot of patients are just, you know, you're suffering for a year because the medication is not working. So the doctor is kind of annoyed because you're like the only patient who doesn't tolerate, and you know, you get the explanation. Sorry, here it doesn't work, but change the medication, it does work. And for example, this Plavix example, it's an enzyme called 2C19. And if you're a poor metabolizer, the drug is not activated. So you get a stent, you pay for the drug, you take it for a year, but you don't have any efficacy. So then you get another stent, which costs, I don't know, at least 5,000 francs for something which could have been prevented. Yeah, this shouldn't happen. But the legislation and the new law they issued, the genetische Untersuchung, it's really progressing. So they even say it's in certain cases you can do the testing before receiving the drug. So it's changing, and for the better. And they're really trying to keep up the pace to be with the current research.
Okay, very good. Yeah.
Just from a practical perspective, when you need to get some medication, probably a little bit under time pressure, would it make sense to do this test early before you need any sort of medication? So whenever you need a medication later on, you already know where complication could occur.
This would be the best, because then every doctor who is expecting, oh, this could be, I mean, they're well aware about the risk. They could ask, like the pharmacologist, is it okay, this drug here in this patient? Would be the best case scenario. But right now it's not reimbursed by health insurances, but they're very modern, very progressive ones like Hasana, which are thinking about, listen, maybe we should do like a special health insurance product, which we test the patient in advance, and we, you know, try to be preventive. We're trying to figure out before he takes a medication to prevent these cases of serious side effects.
As we are talking, it occurs to me, is that only relevant to medication, or could there be an effect on food, for example, as you're talking about health?
Yeah, food is a major thing. For example, Abu Dhabi, very rich people, they want to live forever, like we all do. And they don't need any medication because they do a lot of sports, but nutrition, they like really doing a lot of research on nutrition. But compared with medication, where you did like a lot of pharmaceutical research, the data is lacking. And for nutrition, you don't have the business case, right? The Migros won't sponsor a placebo-controlled trial in eating something and measuring your genetics. And unfortunately, I think they should. And often nutrition is something you don't find the effect in the genes because we don't know all the pathways, but often you can do metabolomics. You can look at pathways and see how efficient are the nutrients available, right? And you get like a hint what is missing in this patient. So it's a current research. If you go to the machine learning center in University Zurich, they all work on trying to figure out having nutrition as a factor with genetics and trying to figure out what is the best nutrition for you to keep you healthy. But spoiler alert, sports is like right now the major factor currently still.
Actually, I mentioned it just today. There are three things which you cannot delegate to somebody else. It's movement, it's sleep, and it's food, basically, nutrition. And to handle that properly, you need a mental health on top of it because you need to be disciplined enough to do it properly.
Yeah, but I would advocate having coaches. I mean, there are very expensive medication, and maybe just a personal coach which helps you with your training has a similar efficacy. But again, you don't have a business case for the coaching, but you have a business case for medication. So we should be more open. And this is a trend we are seeing with these models. If you kind of predict outcomes, you start to look at different outcomes, like what's the impact of nutrition here? And because machine learning is able to consider so many factors, this nutrition, sport, coaching becomes more interesting, and you see the effects easily, more easily.
So one question about these studies, all your models are obviously trained from these studies. Just my layman's question, these studies are publicly available? Are they published by the pharmaceutical companies?
Yes, yes.
So everybody who knows
studies, we could do more, but then we need their private studies, which we are not getting at the moment. But yeah, and this is just the clinical studies they published, right? So it's a limited set, but it's the available set, yes.
Okay, and
another thing is what the modern approaches to machine learning. These methods are so capable that it's even possible that like you pre-train your model like we did right now, and then every time we see interesting patients, we measure like their parameters, their exposure change. And in the models, it's possible to do kind of real-time training to kind of, if you have one patient is enough. You just put the weight, this is only one patient, it's not a big study. And the model will pick up, does this one patient comply with all the others things I predicted, and is trying, and you can see if it does work in the model. So like every patient counts. And if you get to the point where all the saluto nodes measure and have effects and share the data, we can use the data from every single patient and person to actually retrain the model a bit further.
are you doing that regularly? How often are you retraining your models?
I would like to do it more often. Our model trains in 8 hours on a standard, pretty good computer. We could make it much more complex. Then we would need access to better computing power, which we did in the past, but then you have this retraining problem. But yeah, we're working on that. So our models gets more complex, so the training gets bigger, but and it's also a trend that you're considering different sources. And it's not just like the drug-gene interaction studies, you also try to consider in vitro studies from the lab. And so it's a trend to, it's just like the large language models. They just go petabyte scale on data, and with the machine learning and the digital twins, you kind of have the same effect. The data you ingest to use to train the model gets bigger and bigger.
I'm just thinking, are you giving lectures on universities to train people how to train the models?
Not really. Sometimes I'm invited in different countries to be a lecturer, and like my pitch is actually I build the infrastructure because I help you build the infrastructure, because we realize genetic data is very important to governments and to the people in a certain country. So you don't share genetic data actually cross-border usually, okay? So it's very important for a lot of countries to build up their own clinical intelligence machine learning units. I mean, here we have ETH, University of Zurich, and we have really great researchers, but in other countries, they don't, right? And they're hiring a lot of researchers right now. But it's kind of always this, it's a similar problem. And we're trying to, help them build the infrastructure, which actually works, because it's always different for your population. The medication is always different. It's always different, the specialists who are prescribing the medication. So I guess it's a different product kind of in most of the countries. So
So that the procedure to get there is quite similar, but that the outcome is most likely different.
Yeah, because right here, it's like we will excel if we share data and contribute to retrain the model. And it shouldn't be like one company having access to the best model. These models should be shared. There should be research improving the model. It should be very open, like contribution from all sides with these models. And this is the way to go. And this will be different in each country.
As Marco said, we need all kinds of different data to visualize this multidimensional space of personal health. And genetics, genomics is certainly one data track. But what it also requires is the human at the center, because he's the maximal integrator of his clinical data, his genomic data, and his mobile health data, including food data. So that's why I think any transformation that we see towards value-based healthcare, what matters to you rather than what is the matter with you, we need to start from the individual, and that's the message basically of our association. So if you look at the current initiatives, DigiSanté with this Wimmelbild, you don't find the human in here, right? And with EPD, we don't know what the benefit for the individual is. This is the problem. And we want to show the benefit for the individual. We want to empower the individual here at the center. So this is the MyData slide, basically, Pete, that is the aggregator of all his data and can share the data in a health data space that is fairly administered, etc. So the vision of our association is not to build new infrastructure, but to raise awareness and collectively set up the boundary conditions to make this work. When we talk about data, I like this slide where we go from sort of the historical perspective of how data is recorded to the present day, where every doctor has their patient files, and maybe they give a copy into the APD. We have to go into a shared medical record, where the data is recorded once in a structured fashion and separated from any application, so that it can be visualized independently for a patient or for a doctor. And so this is the integrated health dossier, or the APD 2.0, where the individual has absolute control of who's accessing this data. So we can preach this and write white papers, or our idea is really to show that it works. And this is why we decided to do an expedition into health data space. We want to take like in an expedition to outer space, we recruit astronauts that are motivated and educated to do that. We are recruiting salutonauts and mediconauts, just individuals like the ones that are on this call, who say, yes, we want to participate, we want to show that this works. And so our expedition goes, each individual collects, requests their own data. We help them to restructure data from PDFs. They keep the data by themselves. We're interested in their experience, not in the data in the first go. So each of us undergoes a structured health check from a medical doctor, but that results in 7,000 structured data points. We request our health insurance data, our vaccination data, mobile health data, and laboratory data. And with the help of companies and machine learning, we will structure these sort of sorted data in a way that every one of us has such a set of data in the end. And as I said, the aim of Expedition 1 is to know how can we get this data? Can we store it in an EPD or in a personal data store? And what is the experience that each of us experience in doing that? All the salutonauts are connected through a logbook in which we not put the data, but the experience of what was it to open an EPD, to request the insurance data, or, for example, to request your genomic data from 23andMe or any other company. And only in the second phase, we would then use these data sets, and each individual salutonaut decides, yes, I'm going to share this data anonymized in and use these data sets to test health data infrastructures. And collectively, we define the conditions for the trustworthiness and the security and the responsiveness of these health data infrastructures. So the first conclusions is we are really the first beta tester of the EPD. It is the first real-world data for health data infrastructure from the point of the individual. And we see that already with some 40 or so participants who have requested their data, have opened personal data accounts, there is a lot of feedback, a lot of pressure to the providers to make it easier, because currently a health insurance company has to spend at least half a day to collect all my data, and they don't get it complete. And if thousands of people come and request this data, then they're out of business just for that. And so they're coming to us and saying, how can we work together to make this easier? So it's a bottom-up movement that can expand, as we say, to other sources of data, including genomic data. And I think our salutonauts are a great beta tester team that is motivated to go this way, and it would be a great way to start a movement also in pharmacogenomics as a sort of personalized health service. So this is the pitch that I have. This is my digitale Gesellschaften slide, similar to what Pete showed at the beginning. So we have the Chinese model, which is state-centered, where the state sucks up all the data. We have the surveillance capitalist model, where Google and GAFAMs sucks up all the data and monetizes the data. And our vision is that in Switzerland and in Europe, being way back, we can use this citizen-centered model where we have control over the data and can aggregate more data than all the other models. So we have different partners that were also present at the second GDS forum in Olten in June, that help us to sort of structure data that is not structured yet, or integrate data and use large language models. So that's what we want to do, and I think in the discussion we can clearly see how we can expand this also to other data sources like genomic data that Marco was talking about. So
thanks to ETH Zurich, Helzana and the University of Zurich, we have access to great research and machine learning expertise in Switzerland. And this enables us to create really new clinical intelligence we never thought was possible. And from a data science point of view, a patient's health is just a multidimensional optimization problem. And however, to understand all dimensions of the problem, additional data is needed, like genetic information. And we are already able to use genetic data to find patients at risk with their prescription data. And we're able to understand why people respond differently to drugs. And therefore, we should focus on helping doctors and pharmacists more to get more practical access to this new form of clinical intelligence. And because less trial and error to find the right drug means greater patient safety for all of us. I think we already started the journey, and to reach the objective in 5 years to have more patient safety, we just have to enjoy the journey and try our best to create as much clinical intelligence as possible. Every data and every model counts to make better decisions.
This concludes today's joint MyData Zurich and DIDAS Health webinar. Many thanks to our guest experts Marco Egbring and Ernst Hafen for their insightful presentations. Don't forget to check out our show notes for links to more information. If you found the content useful, give us a like. Do it now, I'm waiting. And if you don't want to miss future episodes, consider subscribing to our channel. Thanks for watching and see you next time.